are usually measured in moles, where 1 mole is equivalent to 6.02 multiplied by 10 to the power of 23 molecules. And this is the Avogadro number and the unit of Dalton, which were defined such that this number will be equivalent to 1 gram. Then there's molarity, molarity, which is the number of moles of a solute per solution. We're delving a little bit into chemistry here. Sorry for that. And possible evolution of life on other planets. Yes, this is a very good question. Biologists seeking life on other planets have concentrated their search on planets that might have water. More than 800 planets have been found outside of our solar system. There is evidence that few of them have water vapor. In solar system, Mars have been found to have water. Yeah, which is here and there, okay. And the acidic and basic conditions affect living organisms. Yes, definitely gonna find this in one of the questions. Hydrogen atom in hydrogen bond between two molecules can shift from one to the other. Hydrogen leaves its electron behind and is transferred as what? As a proton, which is a hydrogen ion. The molecule that lost proton is now a hydroxide ion. Hydroxide is the molecule that lost its proton. Hydrogen ion is the proton that has been transferred to it. And the molecule with an extra proton is now hydronium ion, and it's indicated by this symbol here. Through it, it is often represented as also as H plus. Water is an state of dynamic equilibrium in which water molecules dissociate at the same rate at which they are being transferred. So whenever they ask you about the equilibrium, they are talking about mostly a state which the reaction is in the same rate or is moving at the same rate. And mostly you see here when it's being indicated by the forward and the reverse question right there, which is then indicated. And through statistical error, the dissociation of water molecules has a great effect on organisms. The changes in the concentration of hydrogen ion can drastically affect the chemistry of cell. The concentration of these two molecules are equal in pure water, and adding certain solutes called acidic and bases, which modify the hydroxide ion and hydrogen ion. Biologists use the pH scale to describe whether solution is acidic or basic, the opposite of the acidic, which is the base. So acidic acids and bases. An acid is a substance that increases the hydrogen ion, and the base is a substance that reduces the hydrogen concentration of a solution. Strong acids and bases dissociate completely in water. Weak weak acid and bases reversibly releases and accept back hydrogen ions, but can still shift the balance of the hydroxide and hydrogen ion solutions away from neutrality. So the scale is that in any aqueous solution at 25 degrees Celsius, the product of hydrogen and hydroxide is constant and can be written as this particular equation here. And the pH of a solution is defined by the negative logarithm of the hydrogen concentration, written as the formula here. And for neutrality, the aqueous solution has to follow these patterns here. But what you need to just the gist of all of this is summarized in the statements here, that acid solution have pH values less than 7, and basic solution have a pH value greater than 7, and most biological fluids have pH value in the range of 6 to 8. And this will just be your example of the scale in which your pH scale is in mostly of the known food substances. Think of what you have eaten already. You see acidic solution. Yeah, it'll be your battery acid, your cola here, your What is this? What is this fruit? It's orange. Yes, orange. And then this will be your basic solutions. We once talk about buffers. The internal pH of most living cells is close to 7 buffers of substance that minimize the changes concentration of hydroxide and hydrogen. In a solution, most buffer solutions contain weak acid and corresponding base which combine reversibly with hydrogen ions. And acidification is acidification, which is a threat to our ocean. Human activities such as burning of fossil fuel threaten water quality. Carbon dioxide is the main product of fossil fuel combustion, and about 25% of human generated carbon dioxide is absorbed by the oceans, only by the sky, which many people carbon dioxide dissolve in seawater from carbonic acid. This process is called ocean acidification. And this is your example of how we contribute to generate the carbon dioxide which is consumed or which end up there. Right here, what is called ocean acidification in this regard. So this will be your seawater acidified hydrogen ions, which combines with carbonate ions to produce bicarbonate. Bicarbonate is required for calcification, which is the production of calcium carbonate by many marine organisms, include reef building corals. We have made in that regard, which ends up causing those kind of ocean acidification. And this will be your example of some that you don't need to worry about this, where it's just an indication of calcification rate and the gram or the measure of seawater in this regards of the collection that they have used. This will be your acidic donate hydrogen in aqueous solution in just a formulative format in that regard. Any question?
I do have a question on the definitions of specific water specific heat or high specific heat and the definition of a calorie. Are they this? Is it the same definition? So that is the calorie one is saying is the amount of heat required to raise temperature of 1 gram of water by 1 Celsius degree. And let's go to the water one, the specific heat.
You mean the one for water?
For specific heat now.
Specific
High
heat?
specific. There we go.
Okay, the specific heat. So between the calorie and the amount of heat which is required to raise temperature. So this will be how your calorie it's required. So it's the measure in which if these calories is required to measure this kind of temperature. But in terms of the specific heat, is substance which is the amount of heat that must be absorbed. But here is the amount of temperature that can be required to raise the temperature. But I know the required and the one that's, this is the act in which the energy which will be doing the work, which is going to be exerting the power, which is going to be doing in terms of calorie. But for the specific heat, you just have to absorb for the substance in which that 1 gram will be able to change that temperature by 1 degree Celsius.
Oh, now I get it.
Sometimes I'm saying the same thing. But
sorry, sir.
Yeah.
Sorry. I don't know if maybe I got his question. The way I'm understanding it is like, since how the definitions are similar, because for all of them it's the amount of either the heat that's lost or the heat that's gained, right? I think that's because like calories, like isn't specific heat is measured in joules, right? And then the joules can be converted to calories. So they're kind of like the same thing, but with just conversions between them.
And here, if you check the formula, it says the specific heat of water, which is 1 cal, and which is gonna be converted or per gram of that particular degrees Celsius. We state here for this will be per gram of that substances, which is gonna be changed by 1 degree Celsius. And from what I'm understanding here is that I think this is not a fixed number. If you can get this specifically for 1 calorie that.
Yeah, I do understand. So thank you so much. Thank you, Chindebele, also for the explanation.
Yeah, it's really
You're welcome.
similar. I understand. Thank you. It's really similar, but yeah, then it's last year we had the issue with these atomic numbers and all that. They also definition also they are very very similar and sounds alike. But yeah, calorie in that one. Any other question?
So I'd like to ask if I'm making the right conclusion here. So the buffer, are we referring to an amphibole, right?
When we said the word
Buffer.
buffer.
Yeah, are we still referring to an amphibole? Is that the other word for it? Or is it something else?
I'm not sure. The ungulate. What can you spell that?
Amphibole. Oh God, do I still remember how to spell it?
Just type it on the chat box.
Amphibole would be AMPHOLYTE. Because it's giving the same definition as what we were taught in high school when they were referring to an amphibole, a substance that can act as either a base or an acid. Would that be the right conclusion to deduce that? That's what a buffer is.
It should be because there are some referred, but for the benefit of this here on your notes, in which the tone of the examiner will be, I will just recommend that you just stick to the buffer.
Okay.
But
Or,
I know there are. What was the spelling again?
if I'm not getting it wrong, I believe it's AM PH OLY LY. No, POLYTE. POLYTE.
TP.te.
Yeah.
I'm polite.
Yeah.
All right. Can we go to the quiz or any other question? Okay. So we're still going through how you should tackle your questions. These are not notes. It's just for me. Take them, don't take it. Yeah. So this will just be an overall generality. All the stuff that you will see on your question paper, number your question correctly as stated in the question paper. Give answers that are relevant to what is being asked by the question. Questions like distinguish between or differentiate between ones, you could give the differences between the two concept of things. Distinguish among means you must give differences among several concepts or things which are usually more than two. Describe means you define the concept of the definition of a concept or a thing, you should be in detail. Briefly describe means short description of a concept or a thing, which for your benefit most of you won't need to worry about for the sake of your exam. Then we were stopping here last time on the ribosome, which are made up of a large a subunit of small parts of the subunit. I'm gonna need to scoop my slides here. Then there will be growing peptide, there's amino acid, ribosome, lines, units, RNA, transfer RNA and messenger RNA. And then there's the mitochondrial matrix, which is crystal at outer membrane, inner membrane here, where they say this electron micrograph shows the mitochondria as viewed with the transmission of electron microscope. This was viewed in electron microscope. The inner membrane contains four crystal, which increase surface area. So the importance of reading the description of a diagram is that you also find the function of these organelles, which is very significant because sometimes your examiner may just ask you a question based on the function or the definition of these organelles. The space between two membrane is called intermembrane space. The space inside the inner membrane is called mitochondrial matrix. ATP synthesis take place on the inner membrane in that regard. What does ATP stand for? That's an easy question, or that's a boring question.
Adenosine triphosphate.
All right. Then within the human body, certain cells within different tissues may have abundance of one particular organelle due to the function of that cell. If there is a cell in the body which needs to produce a lot of protein, then what organelle might have the abundance of what?
Ribosomes.
Anyone else?
It's mitochondria.
I concur with ribosomes.
Okay, with ribosomes. Okay, then you guys are correct because ribosomes are the organelles which are responsible for protein synthesis. They take instruction from the nucleus and they use them. They use them to take instruction from nucleus and manufacture protein from the cell. If a cell is found in a place that requires the production of more protein, then it will often possess abundance of ribosomes. And somebody read the second question. If you read the second question, then you are exempted for giving an answer.
The answer is Rob runs across the country for his high school. Rob is able to run long distances with the help of the muscle cells in his body. Which organelle in his muscle cells is responsible for helping him finish his long runs?
I think
The
it's
answer?
the mitochondria.
It's
mitochondria.
Mitochondria.
Yes.
Does anybody has you can say no or yes? Does anybody has an idea why or an explanation or know how to tackle if they said also provide a reason why?
I think because mitochondria provides all the power or the energy that is needed by cells, since it's the powerhouse of the cell. So it will give the power maybe to the muscles when he is running so that he is able to run a longer distance.
Yes, it's mitochondria. Why? Because in his muscles, so whenever you want to give an answer, try to chip in or hold in what they're talking about, not necessarily what mitochondria is or what the structure of the mitochondria is on the other side, but correct the mitochondria in his muscle provides energy in the form of ATP, which allows him to run for long distance. So when his muscles cells begin to receive less and less oxygen in his run, he therefore continue. The mitochondria begin to partake in aerobic respiration and produce reduced amount of ATP along with lactic acid. The mitochondria is known as the powerhouse of the cell in this exact reason, and we know that. And Dominic is a doctor. Yes, Dominic. In the ER, patient has just been admitted with possible alcohol poisoning. Ile poisonana, mugruvun. Aye. which organelle within the cell in his liver are actively working to try and get rid of this poison?
It's the Golgi apparatus. The Golgi apparatus is, I think, yeah, the Golgi apparatus.
Okay.
I think it's peroxisomes, the last one.
We have different options now.
I don't remember.
In 3, 2, 1, is it either Golgi apparatus? So it's peroxisomes. Peroxisomes are found around membrane-bound organelles which are filled with enzyme, and these organelles are able to break down fatty acids, amino acids, and toxic substances. For this reason, we have a high amount of peroxisome in our liver cells. So this patient Dominic is attending is likely overworking with peroxisome in his liver cell, which has been overloaded with alcohol. All right. So a chloroplast, here you see here this the structure of a chloroplast, which is an organelle that is only present in plant cell. It is a plastid that contains chlorophyll and is also where photosynthesis take place. However, chloroplast is not present in all parts of the plant, and therefore you see then I also put this also as a structure. We're gonna have to check also the organelle and stuff like that. But the question here is, in which part of the plant is chloroplast not present?
I would say roots.
I didn't get that. You said what?
Yes, I also go with roots.
All right. And indeed, he is right. So chloroplasts require sunlight in order to function. If the roots are in the soil, sunlight cannot reach them. So chloroplast will not be present in the roots of the plants. That is also why many roots plants don't appear green. So in any case, this question can also suffice, and they say why, explain why most of the roots don't appear green or don't appear to have the green color. This is the explanation which has to do with photochloroplast. Then this is just a differentiation of the structure where you see here the A will be a typical animal cell, B typical eukaryotic plant cell that has cell wall, chloroplastids, and the central vacuole structure which is not found in animal cell, and plant cells do not have lysosome and centrosome. I just put this structure so that you can be able to have a distinctive description on structural differences. You see here, this is the plant cell. This one is an animal cell where you see here the nucleus and all that, the cytoskeleton, the microtubules and their function and their description, nucleus, which is in such region where ribosomes are formed, peroxisomes, where metabolizes waste, and the plasmic reticulum, which is associated with ribosome, which makes secretory and membrane proteins, smooth one which makes lipids, and also here plastids which store pigments, and chloroplastids of photosynthesis, cytoskeleton here in the plant cell, which are microtubules, intermediate flagellates, and also nucleus, which contains chromatin, a nucleum envelope, and a nucleus, and in animal is also in animal cell. So go through this and check the differences between these two structures in that manner. Then anybody also read the first question?
Vivica is looking through a light microscope in her biology class and is asked to identify the cell on the slide as either a plant or animal cell. Which of the following organelles present would help her identify the cell?
Do we have the answer to the question?
I think the answer it
Is
will
the
be
presence of centrosomes.
Okay, centrosome. And
Yeah.
you, my sister, wanted to go first. You wanted to go for what?
I was also going for centrosome.
Do we have the same? Did I hear also somebody say centrosome?
Yes.
All right. Let's check the verdict. Yes, centrosome. Because centrosome are organelles only present in animal cells. They aid in cell division. During cell division, the centrosome will duplicate and aid in the process of dividing the cell's genetic material. Where through the plant cell also they duplicate during cell division. They do not contain the centrosome, but the presence of this organelle will help the Vivica into identifying the cell as an animal cell. Plant cell contains specialized organelle which allows them to harness light energy and to turn it into chemical energy for the cell. This organelle helps differentiate plant cell from animals, which organelle is being described. Hence, I put and animal cell. What is the answer? Is it mitochondria, chloroplast?
The answer
Chloroplast.
Okay. Anybody else?
I also go for the chloroplast.
You also go for chloroplast.
Yes.
You guys are ready for examples? Yes, it's chloroplast because they help carry out photosynthesis, where light energy is turned into chemical energy for the cell. So, having studied because you guys have studied, you know they are talking about conversion of light and energy to chemical energy, and you know one of the reasons for chloroplast is that they carry out light energy which is turned to chemical energy for the cell. And plants are the only cells that contain chloroplast, and this organelle filled with green pigment, chlorophyll. That's how plants get their green color. So you already know that. So a tricky question like this, you will never be misled to think otherwise. All right. Anybody to read this one?
Jamie is a chemist with an exercise supplement company. He is helping formulate a new protein powder which includes essential amino acids and is looking for the best amino acids that will readily dissolve in water to produce a smooth protein shake. What part of an amino acid structure should Jamie be looking at to determine its chemical nature?
Thank you for that. We have an answer to her question. These are case studies, by the way. They just help you to just not to know what is R group, what is amino acid, what is carboxyl group, but they also are helping you to have the gist and the understanding of what are they do in the case study. So anybody who have an answer or you think you got an idea what the answer could be?
I think the answer is the amino group.
Amino group, anyone?
I think the R group, the R, the third one.
R group. Oh, I didn't put the numbers on the answers. Okay, I see. Yeah. The R group. Somebody else. We have two different answers now.
I also think the R group.
Okay. The R group is clustering now. And another support for R or amino group. In 3, 2, 1. Okay. I think the R group, guys, you are correct. Why? Because the R group of each amino acid is different, and each R group has specific properties that determine the function of that protein, which is part of Then the second one, it says proteins have many functions throughout the human body. Which of the following is not a function of proteins in the body?
I think it's transport substances in and out our cells.
I think it's
I
the
think the storage of genetic
breakdown of
information.
carbohydrate molecules in the stomach.
Okay. Then my
I think
sister, you were saying
the storage of genetic information.
we have now 3 different answers.
I'm
Anybody
also,
else?
I'm
Yeah,
also going with the
also.
storage of genetic information.
Okay. Now we are clustering in the genetic information storage. Anybody else?
Hey,
Storage of genetic information.
I'll also go with the storage of genetic information.
My kids, we have a last say. You wanted to go?
I don't have an answer, say, but I think DNA is a protein, so proteins they do store genetic information. I'm just doing elimination here. Then the breaking down of carbohydrates is done by the amylase, and all the enzymes they are proteins. So that one is out also. Then the transportation. I'm getting confused between these two now. The contraction of muscles that cover our skeletal system and the transportation of substances in and out our cells. I know that there are some proteins which transport some particles in our body. For example, the hemoglobin. It does transport, but now I don't know. Does it get into a cell? The last time I checked, I think it doesn't get into the cell.
No, but for the transportation in event, there are what you call the channel proteins and carrier proteins that help to move substances in and out of the cell. So that one
Okay.
becomes.
So, which means we will be left with the contraction of muscles that cover our skeletal system.
Can I just give the answer? Or do you wanna just have a I like the idea that you're doing the one for eliminating the wrong. I always say sometimes it's not about knowing, sometimes it's about eliminating the answer which is wrong. I feel like it's not gonna be fair.
Sorry. May I ask the previous say who was saying you were saying with the so we are being left with the contraction of the muscles that cover our skeletal system, right? But aren't muscles built up by proteins? Yes, I'm saying he was saying by process of elimination, he is leaving the contraction of the muscles that cover our skeletal system. But then I'm saying, isn't muscles are made up, they are built up from proteins.
Then if they are built up from proteins, so you are also coming here with the further elimination, the contraction of the muscle, because it won't be falling under the category of what we are looking for, because what we are looking for, we are not looking for something which is of a function or something which, because if it's built up from protein, sorry, what do you know? Maybe I also have a flu. Oh. But what we are looking for is what is not the function of the proteins, or what is not part of the proteins, what is not helped build up by proteins. But I like your approach, and I like this approach of going through. I'm not thinking maybe we should also go through the each and every question, not each and every question, but each and every option. Why is not the correct answer? I didn't include this in this note, but in the next one, I'll go through so that we can be able to dissect why this option, because we also only explain that for this one, we only explain that because it stores our genetic information, and we explain why it stores genetic information through proteins. Although they do many things in our body, they do not store and our genetic they do not store our genetic information. This is done by other biomolecules called nucleic acids, which make up our DNA system. So here you need to know two things here. You need to know what stores our genetic information. And you also need to know what made up of what mean proteins. And then you need to know and be able to, this is about, he's right. This question is about eliminating what is not the function of a protein. And that is how you should be able to deal with this kind of questions.
May I? Sorry, 1 second. Saying I think that time my phone kind of cut off when I was saying like by his like process of elimination, right? He's saying what he was saying that DNA is a type of protein, but I don't think DNA is a protein. Like DNA helps with information on the proteins that are to be synthesized, but it is not a protein itself. And the nucleus with the DNA is the one that contains all the genetic information and the activities of the cell.
I will try to bring these kind of questions in the next session also, and also I will try to also not only explain the right answer, also why but this one, the approach is that chances that 90% of the 4, unless if they say none of the above, there's an option. Chances that the 3 are the function of the proteins in the body. So which one is not the function of a protein? So here we talk also about the structure and functioning of carbohydrates. We're going to talk about the carbohydrates. Carbohydrates can be presented by, I also include the notes here inside so that they could help you when we tackle a question as it goes on, and also this is the formula. So it's dynametric formula, where the N is the number of carbons in the molecules. In other words, the ratio of carbon to hydrogen is 1 ends to 2 ends to 1 in carbohydrates molecule. This formula also explains the origin of the term carbohydrates. The components are carbon, which is from carbo, and the components of water and hydrates. Carbohydrates are classified into 3 subtypes: monosaccharides, disaccharides, and polysaccharides. And the monosaccharides, mono, 1, saccharide, sweet, simply sugar, the most common, which is glucose. Stay away from sugar, by the way. In monosaccharides, the number of carbons usually range from 3 to 7. The monosaccharides names end with the suffix "-ose". So if the sugar has aldehyde group, the functional group, which the structure of the RCHO is also known as an aldehose, and it has a K-note group, the functional group, which is structure RC. It is also known as a ketose, depending on the number of carbon in the sugar. They also may be known as trisose, 3 carbons, pentose, 5 carbons, and hexoses, which is 6 carbons. We'll also go through this thoroughly. I will try next week so that we just get the gist of the notes for this, because I feel like I'm just summarizing what could be a very, very significant topic here. And this is just a structure of the glycosaccharide, and this one here was ketose, aldose, and trisose, pentose, and hexose. The chemical formula for glucose here, as we already know, and in humans, glucose is an important source of energy. During cellular respiration, energy is released from glucose, which is that energy which is used to help the ATP. Plants synthesize glucose using carbon dioxide and water, and glucose in turn is used for energy requirements for that plant. And excess glucose is often stored as starch that is catabolized, which is the breakdown of larger molecules by cells, by humans and other animals, respectively. So we'll also go through this and also go through that also about the monosaccharides, structure of function of carbohydrates in your notes. And then they said Sarah studying nutrition, and as an assignment she has been asked to find example of each type of carbohydrate in the food that she consumes over the course of the week. Sarah is eating pasta today and knows that it contains starch made of a long chain of many sugar. What type of carbohydrate will you classify starch as?
Polysaccharide seconded.
Yes, it's polysaccharides.
Yes, okay. Polysaccharides are complex carbohydrates that are made of long chains of monosaccharides. And we did say that poly means many, and stated starch is made of many polysaccharides. So long, many, more monosaccharides, which are linked together in that regard. Then there's Malik, has a huge trek meet tomorrow. Malik should focus on eating food rich in what type of macromolecule? Select the best answer. So here, when they say select the best answer, answer will be wrong, but we just want to select the best answer.
Hi, so
But for
um,
it will be the right answer.
since
Yes, how are you?
protein, carbohydrates, and lipids are all molecule. I mean macronutrients, which means they're made up of macromolecules. How are we answering?
I think it will be carbohydrates since he is
No?
running, he needs energy. So basically the micromolecule that he needs to get energy from will be the carbohydrate.
Oh, okay.
Okay, just hold up. But I think the
There's Mike that
question,
is making noise.
the question
Is it just me?
was can you
Is it just can't hear you, sir? Because I can't hear
can't
you
hear me?
cutting. No, I couldn't hear you at all.
Okay, no. So okay, can you hear me now?
Yes, I can.
Yes. So what was your question? Sorry, but I couldn't get you also.
Yes, my question was, since
And this every okay.
micronutrients are protein, carbohydrate, and lipids. So I wanted to
Mm-hmm.
ask, since they're macronutrients, they're made up of macromolecules. So how are we going to choose the best one?
May I?
Yeah. Okay. Okay. Go. And
I think
also,
Uh,
we look
Hello?
at the function that they need the macromolecule for. That's what's going to determine what macromolecule they need. So since they have
Hello?
a huge track meet, they are running, which means they need a lot of energy. So carbohydrates are the ones that give most energy. So the macromolecules would be carbohydrates.
Sorry,
say you
I
want to
what?
give
couldn't
it a
hear you.
You couldn't hear
Sorry.
me.
step,
Oh, no, I couldn't hear you.
I was saying how
but
we choose is we will select depending on what you look at what they need to do or what they need from those macromolecules to choose from, right? And since they are going to run, which means they need more energy. So them needing more
Mm-hmm.
energy is the macromolecule that gives us more energy is carbohydrates. So I
Yeah,
think the answer becomes carbohydrates.
okay. Thank
I
you.
do. I also second with her with the idea that for the sake that the person is going to need more energy, so carbohydrates is the macromolecule that you need to take in. But now we also have lipids. Lipids, they also produce a lot of energy. For those who are doing nutrition, I think they know that lipids, they can, one gram of lipids produce about 38.
It's 13. No,
And
it's 38. It's more.
it's more. But now, the reason why I'm seconding for carbohydrates, it's because carbohydrates, they are simple to digest. The energy to extract energy from carbohydrates, it's a simple process as compared to extracting energy from lipids and proteins.
thank you guys. I like this also when it turn into a group discussion, because that's, trust me, you will understand more when you are engaging one another than somebody like myself, just yapping then what. But yeah, carbohydrates provide your body with quick energy that is readily available to use. So Malik will be exerting a lot of energy on his track to be the best choice for him. Explain the differences between cytokinesis and plant and in animal cell. So you have to explain in animal cell, in plant cell. So in animal cell, cytokinesis occurs by a process known as cleavage. The first sign of cleavage begins with the appearance of cleavage furrows near the metaphase plate, which deepens until the parent cell splits into two. And in plant cell, have cell walls which form no cleavage and furrow, and vesicles from Golgi apparatus collect the metaphase plate and the vesicle fuse with each other to form the cell plate. The cell plate closes with fuse with the plasma membrane in that regard. Just go through it when you get hold of this. Distinguish between asexual and sexual reproduction. I think we have done this.
Asexual reproduction is it has only one parent. There's only one parent, and the offspring is identical to the parent. And then in sexual reproduction, it needs to have two parents, and the offspring are not identical to the parents.
Cool. The one parent produces genetically identical offspring in asexual mitosis. And by the way, there's no one answer. So just try to put in the understanding that you know, but you should be writing the correct ones. Obviously, the type of reproduction, two parents give rise to an offspring and have unique combination of genes which are inherited from gametes and the two parents, and gametes are produced through meiosis. List the phases of meiosis 1, meiosis 2, and describe events of characteristic. But can you list them? Don't worry about description.
Yes.
So here is the meiosis 1, meiosis 2, where will be the prophase 1, where homologous chromosomes replicate. Prophase 2 is where the spindle apparatus which is formed. The metaphase 1, where chromosomes are arranged on the metaphase plate, crossing over is completed. Microtubules are attached to a kinome core, and then metaphase 2 chromosomes are on the chromatids of each chromosome, which are not genetically identical because of the crossing over. And anaphase 1 homologous separate. The homologous move towards the opposite poles, where sister chromatids move as a unit towards the same direction. These two sister chromatids at the centrosomes, they are separate. They move individual chromosomes towards opposite poles or in opposite directions. And also in telophase and cytokinesis, each chromosome is comprised of two sister chromatids, where cytokinesis is usually take place in the same time with telophase 1 forming two haploid daughter cell. In animal cell and cleavage furrows and plant cells, the cell plate form, and the telophase 2 and cytokinesis nucleus are formed. The chromosome begins and they decondense. The cytokinesis take place. There's four daughter cells formed as a result of meiotic of one parent cell. Each parent cell has four daughter cells, which is formed haploid and genetically distinct from one daughter cell from the parent cell. So this will be the differences. So while you don't have to worry about drawing the differences, but you just need to know the differences so that if each and every question can come in that regard in terms of differentiation, you can be able to distinguish every question that will be able to come in that regard. All right. I think this is the easy one. What is the basic unit of life?
A cell.
It's a cell.
So,
Oh,
We've done this. Nobody is gonna get this wrong. The basic unit of life is cells. Cells are the smallest structural and functional unit of living organism and are often called the building blocks of life. Cells can perform various functions which is necessary for life, such as metabolism, reproduction, response to stimuli, homeostasis, DNA, organelle, nuclear tissue, that are all important components of living but are fundamental units that makes up this living organism. Nobody's gonna get this one, especially for people who attend. When a base is added to a buffered solution, the buffer will
donate the H plus ions, the protein. Can you hear me?
yes, I did. Ion. Anyone?
The buffer will donate hydrogen H plus ions, yes.
Okay. Do we concur with them? Do we have the final arbiter who will concur with?
Yeah, I do.
Okay, cool. Thank you. Who will donate hydrogen ion because they are designed to resist changes in pH. So when a base is added to a buffer solution, the buffer will work to neutralize the added base by donating hydrogen ions to react with the hydroxide ions from the base, that's maintaining the pH of the solution. These are to keep the solution relatively stable. All right. The pH indicates that
the solution consists of pure water.
solution consists of pure water. Anyone else? That's number one. Yeah, I like that. I like this one. They confuse the heck out of people where they say the option number 5, what they could say it's 1 and 2 are correct.
I also think that's the correct answer. 1 and 2 are correct.
All right. So we have different answers now. We have 1 and 5. Anyone?
I think it's
I-
two.
One.
I think it's two. Because
think
it's
it's
not
two.
that also. I think there are other solutions that have a pH of 7, or other substances that have pH of 7 but are not pure water, or do not probably contain pure water. I don't know.
Okay.
I say it's pure water, which has the pH of 7, because when you say the concentration of hydrogen ions is equal to the concentration of ions, it could be between. It doesn't make sense. Do you know what I'm saying? Because if the concentration of hydrogen ions is 3, how can it be equals to the hydroxyl, since the hydroxyl has to be basic? I don't know if you understand what I'm trying to say.
Sorry, may you please repeat that?
I said the second one, it doesn't make sense to me, since when you say the concentration of hydrogen ions is equal to that of hydroxyl ions, how can they be equal, because they are the opposite? So when the moment the concentration of hydrogen ions is 3, the hydroxyl ions can never be 3, because it has to be greater than 7 for it to be like concentrated of hydroxyl ion, since it's a base.
Sorry, may I? So if you say the concentration of hydrogen ions, this is after you have calculated the amount of concentration that's base, right? So the amount of concentration, let's say we have 2 moles of hydrogen ions and we have 2 moles of hydroxide ions, they balance each other out. So them, because that's H plus and OH minus, and then they balance each other out, and then it becomes neutralized. It neutralizes them. The concentrations are equal. They get to be neutral. It gets to be neutralized, because if it's an acid, it has a greater concentration of hydrogen ions, your H plus ions. If it's a base, it has a greater concentration of OH minus ions, right? So if they are balanced, it means it's basic. It's at 7.
I want somebody else to come in now.
But like,
I just want somebody to come in between the two of you, so that we can close this one. Somebody else.
Yeah, what Tindebele is explaining is correct, because okay, let's take for instance a molecule of water. A molecule of water is made up of an OX molecule and a hydrogen molecule. So a hydrogen ion molecule. So now those two, if they balance, that being said, it means if we do have more hydrogen ions, which is H plus, that solution, even if it's water, it's acidic water. But if we do have an equal balance of H plus ions and OH minus ions, it means we do have a pH of 7, a neutral solvent, a neutral molecule of water. I don't know if it's making sense.
So which answer are we saying it is?
Then the answer is 1 and 2.
It's 1
Water
and 2.
and pH, yes.
So the concentration of hydrogen ions equal that of hydroxide ions. But please go through the slides for the chemistry of water and check on those equations, those functions of the equation, and go through, find an example to go through that in order so that you can have an understanding of if they're not just talking about the fact that the bases are or the acidic formations are, or which one is greater than which element or which value, how is it concentrated into. But for the benefit of this answer, the answer is 2. But through your discussion and your inquiry, I'm also being the solution of consists of pure water. So one of the things that will help you is, in science they always said, shy away from making instinctive or commanding statements which commands you to have like supporting answers. When you said the solution consists of pure water, is, what is pure water now? When we get to that, because the scale of the pH has to be indicated, go through that diagram where it's showing the alkaline, the acidic lines, the strength in which the elements are written into, and how can they be definited into. So can we go through that? Can you go through that? And I'm also putting inside a note and see if we're gonna arrive at a different conclusion, but we do agree and concur with each other that 2 is also part of the answer, right? The question is either 1 or 2 now. Is it fine? Cellulose is I think we have done this. Cellulose is an example of polysaccharide. Anyone else?
Polysaccharide.
Do we have the third option? The third person rather than the third option. Cellulose is an example of polysaccharide, right? Polysaccharides are large molecules which are made up of multiple sugar carbohydrates units. Cellulose is specifically complex carbohydrates that form the cell wall of plants and major components of their structure. And it is not a polypeptide which contains chains of amino acids, making it a protein. Lipids, which are fats and oil. Nucleic acids, which are DNA or RNA or a simple fat. So it's a polysaccharide. An atom is defined as the smallest. I should have removed the smallest, because it gives it away.
Second option.
2.
Okay. I had two. There's also 4 and 5 are correct. Only one person said 2, right? You're right. So the correct answer is 2 particles of an element that can take up part in a chemical reaction. Atom is the basic unit of an element that retains the chemical properties of an element. Atoms can combine with other atoms to form molecules. They also participate in chemical reactions. So option 2 accurately defines an atom in the context of chemical reaction. Shortening is an example of
Cheating.
okay. Not the other, which yeah, correct. Cheating. What is it called? Cheating. Okay.
Yeah.
Thank you. But what is it example of?
Polysaccharide.
Yes, I wanted to say the same thing. It's a polysaccharide.
Saccharide.
No, I made a mistake here. No, I made a mistake. This is not supposed to be there. Can you see that?
Yes, we can.
Do we agree it's polysaccharide, right?
Yeah.
There we go. It's a long chain polymer of glucose and the structural components of cell wall in that. The electron, we haven't talked about the electron configuration, but we did. Of 1s2 belongs to
The answer is neon.
So what's carbon number 12?
The answer is number 4. It's neon. Neon has
Is
10 electrons.
I second that. Neon.
we have another person who will second that. Okay. Yes, you guys are right. It's neon, which corresponds to the element of neon, which is atomic number 10. So the option number 4 is the correct one. Okay, here's the easy one that you guys are gonna get it. Ribosomes are responsible for
The number 1, protein synthesis.
anybody else?
Protein synthesis.
I think you are wrong. Because the answer is, I'm kidding, you are right, protein synthesis. There are still organelles that find site of protein synthesis in cell where they translate to RNA, which is messenger RNA, into amino acid sequences creating proteins. So they are not just they are not involved in digestive system. They are not involved in photosynthesis or controlling the center of the cell or removal of that. It's protein synthesis. So you guys already are familiar with that structure and you know the function of all the label structures there. So you are able to tackle this question as easy as you can get in that order. The mass number refers to, this is the one that we're gonna have. It really looks very similar. Remember that question that our sister asked my brother asked in the beginning of the session. I said they are very similar. They are doing it so that they will bring them here.
Option number 2.
It's option number 2.
Option number
2.
Option number 2.
The combined number of protons and neutrons.
Nah, live blind. I do concur. You guys live blind. Yeah. The combined number of protons and neutrons in an atom. Which statements it's best describe isotonic solutions?
It's option number 3.
I also agree with number 3.
It's number 3. I also agree with number 3 as well.
which is a solution which is with a balanced water and solute concentration.
Yes.
You have answers from somewhere that I don't know of.
I have notes.
Good. That's the perfect answer. Thank you for that answer. That suffice everything. Yes, it's a solution with a balanced water and solution concentration. So in an isotonic solution, it has some concentration of solutes such as salts, glucose, and other dissolved substances as in another solution. In context of biological system, it refers to a solution that has the same concentration of solutes as the cytoplasm of a cell. In such a solution, there is no net movement of water into or out of the cell, which means the concentration of solutes inside and outside the cell are equal, resulting in a balanced state. So there will be a question which says, explain the difference between cytokinesis. I think we did that in plant cell and animal cell. It's recurring. You see here, plant cell, plant cytokinesis, animal cytokinesis. This is just a clear understanding. It usually occurs by cell plate method. It takes place by cleavage. Remember we talked about this previous slides. The spindle usually persists during the spindle and begins to degenerate soon after anaphase. Cell plate grows centrifugally, cleavage progresses centripetally. The new cell membrane is derived from vesicle of Golgi apparatus. New cell membrane is usually derived from endoplasmatic reticulum. The same thing, or the same thing here is the structure which is just showing animal cell. See cleavage, central valve, and then it divides from this and here in plant cell through Golgi apparatus and in all that. So go through all this and put the notes into your notes and all that. You will be able to tackle any question which is going to come after. This ratio 9:3 to 3:1 phenotypic ratio is a characteristic of.
That is number 4, the second filial generation of a dihybrid group.
It's number 4.
F2 generation of hybrid.
Agree.
Of hybrid. Okay. Number 4. Do I have the answer for that? F2 generation. Yes. F2 generation. In hybrid cross, 2 different traits are considered. The F2 generation results from the cross between the 2 heterozygous individuals. Each individual carries 2 different alleles for each of the traits, 2 traits that results in different combination of traits. And for the time, possible combination of alleles, which lead to the ratio of 9:3 to 3:1 ratio. Which statements best describe chemical work?
I think it's number 4.
Number 4 as well, because chemical work is the construction and the glycation of each molecules.
Anybody else? Do we all agree with number 4?
No, I'm thinking of number 5. Diffusion of substances across the selective permeable membrane.
Okay, because we are running out of time, can I just give the answer?
Yes, please.
Pushing of driving is number 4, because the pushing and driving and the genic reactions such as synthesis. Chemical work involve the consumption processes in a cell, such as the building complex of molecules from simpler ones and the genic reaction, which require an input of energy. Can I just also do this one also? Negatively charged ions are known as
Number 4 and you.
number 4. All right, cool.
Number
Correct.
4.
Number 4.
The following term.
It's number 3, evaporation.
Do we all agree?
Very, yes.
I sense we're gonna get a total. This 2024, that's gonna be, people are gonna get total, and they will do what you call it, forensic investigation and found reusers. These kids are just smart. Yes. Which of the following represent a male gametophyte of seeding bearing plants? A, oval; B, micro mother cell; C, pollen grain interior; D, embryo sac; or E, fertilized egg.
I think the answer is C, pollen grain interior.
C.
What did I tell you? They are getting too tough. All right. I think we're gonna stop here for today.